Yeah, thank you very much, Stacy, for the introduction. Also for you all to give us a bit of the background. I think it's always helpful to position a bit how to talk about topics. Of course, we have a lot of kind of slides to cover and a lot of topics to discuss. Hopefully we can make it interactive a bit more than maybe just having the question at the end. We have, at least for the agenda, I can just let you know kind of some changes. So we will exchange the speaker role. So you see there's blue areas which I will cover, as well as black areas which Ryan will cover. So we start a bit of a general introduction, which I want really to set the stage and the expectation also. What is RNA-Seq and really how to analyze it? There's so many open kind of areas where you need to kind of position right and make decisions also during already the data analysis, the sample preparation, and of course later on in the data analysis as well. And we have a very good data set to practice here, which is the IDEA data set, which is from Illumina, issued last year as a competition, which is where we are hoping to win. Of course, it's just submitted, I think, our kind of report for that today, which means we don't know. But of course, it's something where we are hopeful to be kind of also selected as being successful there. But of course, QC and exploratory analysis are the other parts which I like to start with. Hopefully, at that time, we reach the time when the dinner arrives, as we have to kind of be a bit flexible here. That when this arrives, we stop here, make half an hour, focusing on the meal, and then during coffee, I think this is what I understand. So dessert, we continue to talk about it. So slightly, and then Ryan takes over, talks about more the technical aspects like mapping to the transcriptome, creating the statistical model, as we are very proud of having a very powerful statistics, which how we involve that. And there's some kind of fancy things we came around with this IDEA data set, kind of where we want to really get your feedback also how informative, how useful this is, especially because it is kind of the first really replicate data set which we can really demonstrate our abilities to apply statistics to. So therefore it's kind of a showcase also. And then of course for the rest of the talk we will focus on more applications. So gene expression results, of course, the most easy to kind of understand results, but also we talk about alternative splicing, coding SNPs, which you can take from that data in combination with the expression. This is kind of a nice combination, also leading to allele-specific expression, which is another topic on the list. And then, of course, we can also look for novel aspects like detecting unexplained regions by looking for something which is not mapped so far. We will also talk a bit of fusion transcripts, which is a very kind of applied aspect, especially for cancer samples, as these are cancer cell lines we are talking about. I think this is at least an aspect which is interesting. Also, I noticed some clinical background here, so therefore I think this is some relevance too. So let's start moving on. A bit of an introduction about our kind of role, our kind of vision on the next-gen sequencing as a whole. So you see here that there is a kind of the flow of steps here. We will also see for the entire presentation as a kind of in horizontal orientation, as a kind of segmentation of these 7 steps, which you can kind of segment the entire data analysis to. So you start, of course, with the raw reads, which gets delivered from any of the providers which are on the market, like Illumina, AB Solid, Roche, or Ion Torrent even. So all that provides the raw reads, which first needs to be aligned. So the genome alignment is the first step, and also computer intensive, where really a lot of computer power is needed. Also large data is generated and needs to be converted. And then you have the mapping stage, where actually some people get confused about alignment and mapping. So I just want to be clear.